This is part 2 of the MCAT Metabolism crash course. In the previous video, we looked at how individual cells use molecules like carbs or fatty acids to create ATP. Now we're going to look at how the body as a whole regulates those processes and uses some other processes to ensure that all the cells in the body are getting enough of those molecules like glucose and fatty acids to create the energy that they need. We'll look at what the body does in the fed state and in the fasting state. And for each of those states, we're going to emphasize what's happening for these 4 points. In the fed state, blood sugar level is high from all of the food that we've just consumed. This causes insulin to be released by the pancreas. Since we have high blood sugar, glucose is what's being used by the entire body for energy. And in this fed state, we have enough excess glucose that the liver is going to use the excess glucose to create glycogen through a process known as glycogenesis. And an important side note, this process also occurs in the muscles. In addition to glycogenesis, the liver is also going to be synthesizing fats in a process known as fatty acid synthesis. Glycogenesis occurs in both the liver and the muscle, and it's the process of taking excess glucose and connecting individual molecules of glucose into a long chain called glycogen, which is stored in the muscle in the liver to then be broken down back into the individual glucose molecules during periods of fasting when we're not getting glucose from food. The first step in glycogenesis is the same as in glycolysis, where we create a molecule of glucose 6-phosphate from glucose. After that, the phosphate on the 6th carbon is moved to the first carbon of that glucose. And then using a molecule of uridine triphosphate, we attach a UDP to that glucose molecule. You don't have to worry about this, but just to give you some clarity, 2 phosphates are kicked off of that uridine triphosphate during the process. So really only a uridine monophosphate is added, but it combines with the phosphate that's already on carbon 1 to create the UDP that we see on UDP glucose. UDP glucose is then attached to a protein called glycogenin, and that molecule of UDP is kicked off during the process. The molecule of glucose is bound to that glycogenin at the first carbon. We continue to make UDP glucose, and then a molecule of that UDP glucose gets added to that first glucose that was attached to glycogenin. And we call that bond an alpha 1,4 glycosidic linkage, because the bond is between a carbon 1 and a carbon 4. The reason we put the alpha there is because carbon 1 is an anomeric carbon, and any time an anomeric carbon is involved in the bond, you have to say whether it's in the alpha or beta configuration, and this one is in the alpha configuration. And the addition of that glucose is carried out by glycogen synthase. From here, glycogen synthase continues to add glucose from UDP glucose, and every time it adds one, it puts it onto the 4th carbon on the glucose on the end of that chain. It continues doing this, creating a long chain of glucose molecules. Glycogen synthase makes that long chain, and then eventually an enzyme called glycogen branching enzyme breaks one of those alpha 1,4 bonds and takes all of those glucose molecules to the left and moves them to the 6th carbon of that same glucose that it broke them off of. And that bond is called an alpha 1,6 glycosidic linkage. Then glycogen synthase comes back in and extends both of those branches in the same way that we just discussed. This goes on and on until a granule of glycogen is produced, and at the center of that granule is the protein glycogenin, and it has a ton of these chains branching off of it. And a real granule of glycogen has a lot more chains than what I've drawn here, and those chains are longer and more branched. And that allows for a ton of glucose molecules to get stored in that space. So in the fed state, our liver and muscles will fill up with these glycogen molecules. The liver and the muscles have a limited capacity for how much glycogen they can store. And once those glycogen stores are full, excess glucose can't go there anymore. So the liver breaks down that excess glucose using glycolysis, turning it into pyruvate, which then turns into acetyl-CoA in the matrix of the mitochondria. That acetyl-CoA feeds into the Krebs cycle, but at this point, the liver has plenty of energy because we have plenty of glucose, and ATP is an inhibitor of isocitrate dehydrogenase. So because of this inhibition, we have a buildup of isocitrate, which then turns back into citrate. Citrate builds up in the mitochondria and starts to get shuttled out into the cytoplasm. Once it's in the cytoplasm, an enzyme called citrate lyase is going to break it back into oxaloacetate and acetyl-CoA. The acetyl-CoA will then go on to participate in fatty acid synthesis. The most important step in fatty acid synthesis, because it's the rate-limiting step, is carried out by acetyl-CoA carboxylase. And this takes a CO2 molecule and adds it to acetyl-CoA, creating a molecule called malonyl coenzyme A. And again, this is the rate-limiting step of all of fatty acid synthesis. That malonyl CoA will now get attached to a large protein complex that has multiple domains with different names and functions. And the part that malonyl gets attached to is called a acyl carrier protein. And on a separate acyl carrier protein, a molecule of acetyl-CoA is attached. And if you know a lot about fatty acid synthesis, you might know that it's not actually accurate to say that both of those are attached to acyl carrier proteins. But for the depth that you need to know about this for the MCAT, it's totally fine to just simplify it and say that they both are. From this point forward, we're going through the actual steps of fatty acid synthesis. And I want to point out before we start that acetyl-CoA is a 2-carbon molecule and malonyl-CoA is a 3-carbon molecule. And that will be an important point to follow as we go through fatty acid synthesis. The steps of fatty acid synthesis are carried out by an enzyme on that large protein complex called fatty acid synthase. In the first step, the malonyl group is attached to the acetyl group, and during that process, a CO2 is lost. So we combined a 3-carbon molecule to a 2-carbon molecule, but we also lost a carbon there. So we end up with a 4-carbon molecule. And don't worry at all about the mechanism behind how these two molecules get added together. From here, an NADPH comes in and reduces that red carbonyl group, turning it into a hydroxyl group. After that, the OH and this H are lost in a dehydration reaction, creating a double bond between those two carbons. Another molecule of NADPH comes in and reduces that double bond, making it a single bond. So after this first round, we end up with a 4-carbon molecule with 1 carbonyl group at the end. Now every round of fatty acid synthesis is going to result in a molecule that looks just like this, but it's 2 carbons longer. So let's look at what steps occur to make that happen. It's a very similar process to what we just discussed, and it starts with making another molecule of malonyl coenzyme A from a molecule of acetyl-CoA using acetyl-CoA carboxylase. That malonyl-CoA is attached to the acyl carrier protein, and then it's combined with that 4-carbon molecule that we made in the first round. During that combination, a CO2 is lost. So similar to before, we added a 4-carbon molecule to a 3-carbon molecule, but we lost 1 carbon up here, giving us a final molecule that has 6 carbons. And then we do the exact same things that we did before. We reduce that carbonyl carbon to a OH, and then we dehydrate it, creating a double bond. We then reduce that double bond using another NADPH, creating a single bond. And by the end of that, we have a 6-carbon molecule that's just a carbon chain attached to a carbonyl carbon at the end. We continue doing this until we make a long carbon chain, usually about 16 carbons. And at the end of the chain, we have a carboxylic acid functional group that forms once it's popped off of the acyl carrier protein. And that molecule is our final fatty acid. The liver makes a bunch of fatty acids and then starts connecting them to molecules of glycerol. Glycerol is a 3-carbon molecule whose structure is really important to know. And to give you a general idea of how glycerol attaches to those fatty acids, here is a general mechanism showing how the OH on the glycerol attacks the carbonyl carbon on that fatty acid. That happens 3 times and you end up with a molecule called a triglyceride, which is basically just a glycerol attached to 3 fatty acids. The triglycerides are then sent from the liver through the blood to the adipose tissue where they are stored. And then in periods of fasting, when the body doesn't have glucose, it'll break down triglycerides for energy. And we'll talk more in depth about that process later in this video. The last thing we'll talk about is PFK2. And PFK2 is an enzyme that's present in multiple organs, but most importantly, it's present in the liver. Notice that these are the first 3 steps of glycolysis. And what PFK2 does is it gets activated or turned on by insulin, and then it takes fructose 6-phosphate and adds a phosphate to the 2nd carbon, creating fructose 2,6-bisphosphate. And then fructose 2,6-bisphosphate activates phosphofructose kinase 1, which is that 3rd step in glycolysis, and it's the rate limiting step. And this is really important because during the fed state, ATP is inhibiting this enzyme, which is trying to stop glycolysis from occurring. But we need glycolysis to keep occurring in the liver for a few different reasons, including the production of acetyl-CoA for fatty acid synthesis. And this gives the body a way to overcome the inhibition of glycolysis using the hormone insulin. And we'll talk more about PFK2 during the fasting state because there's going to be a different hormone that's going to turn this enzyme off. We're going to break the fasting state up into multiple stages, and the first state takes place 2 to 24 hours after our last meal. About 2 hours after our last meal, our blood sugar drops, so the blood sugar has lowered some. And this causes the hormone glucagon to be released by the liver, and glucagon is going to cause a lot of metabolic pathways to start happening, but initially the first thing it's going to cause is glycogenolysis. And glycogenolysis is going to start breaking down glycogen and releasing glucose into the bloodstream, and that glucose from glycogenolysis is going to be used by all the cells in the body. And remember, the liver was one of the main spots that we said glycogen was stored, so the liver will be performing glycogenolysis. It will also be occurring in the muscles, but the glucose produced by glycogenolysis in the muscles will be used by the muscles, and the glucose produced by the liver will be used by the rest of the body. The first step in glycogenolysis is to start breaking off the glucose molecules from the long chains of glycogen. This is carried out by an enzyme called glycogen phosphorylase, and a phosphorylase uses a phosphate molecule to break a bond. And here we have a phosphate molecule that attacks carbon 1 and gets attached to carbon 1 of that glucose on the end of that chain. That pops that glucose molecule off the chain, creating glucose 1-phosphate. That then gets turned into glucose 6-phosphate and then enters the bloodstream to go to other organs throughout the body. Glycogen phosphorylase will keep doing this until it gets close to that alpha 1,6 bond on the branches of glycogen. And it's actually going to stop once it's 4 glucose molecules away from that bond. So these 4 glucose molecules will not be touched by glycogen phosphorylase. A new enzyme called debranching enzyme will come in and take 3 of those glucose molecules and move them to the end of this chain, forming a new alpha 1,4 linkage. The single remaining glucose molecule is then popped off by debranching enzyme, releasing a free glucose molecule. Glycogen phosphorylase can then come in and start chopping off the glucose molecules at the end of this chain. Part 2 of the fasting state takes place about 24 hours after our last meal, and at this point our glycogen storages have been completely depleted. Our blood sugar levels are low, and glucagon is still being released by the pancreas. A lot of the body will be using fatty acids as a source of fuel. Other parts will be using glucose, which is produced through gluconeogenesis at this point, and some parts will be using ketones. The liver does a lot during this stage. It's going to be using fatty acid oxidation at a really high rate. It will be producing glucose through gluconeogenesis, and it will be producing ketones through ketogenesis. In order for different parts of the body to be able to use fatty acids as a source of fuel, we first have to break down triglycerides. And triglycerides were created during the fed state by the liver and then stored in the adipose tissue of the body. Glucagon is going to activate lipases, which are going to break down the triglycerides into a glycerol molecule and 3 fatty acids. The fatty acids will then get attached to a protein called albumin, which is going to help them move through the blood and reach their target cell. And then once they reach the target cell, they'll get burned up through fatty acid oxidation. The whole process of fatty acid oxidation was already covered in part 1, but basically it just makes a bunch of acetyl-CoA, which then can go into the Krebs cycle to help make ATP eventually. And fatty acid oxidation will be especially active in the liver, and we'll cover why in just a moment. Some parts of the body, especially the brain, can't use fatty acids as fuel, so we have to keep providing them glucose through a process called gluconeogenesis. Since we don't have glucose from our food or glycogen, we're going to have to make this glucose. Luckily, a lot of the steps of glycolysis are reversible, so if we can get one of the molecules in glycolysis, we can go all the way back and create glucose. However, there are 3 steps in glycolysis that are not reversible. So at these 3 steps, we're going to have to use different enzymes than what we used in glycolysis. And as we go through this, make sure you understand that not all gluconeogenesis starts with pyruvate. In a lot of cases, we're actually going to start with a molecule that falls somewhere in the middle of glycolysis. For instance, we might have a molecule of DHAP, and we're going to work backwards from there, depending on whatever molecule we have. If you're starting with the molecule of pyruvate, you will have to get back to phosphoenolpyruvate via 2 steps. First, you turn pyruvate into oxaloacetate via pyruvate carboxylase, and that requires the use of a molecule of ATP. And remember, pyruvate is in the mitochondria, so that is where this step occurs. Once oxaloacetate is made, it's going to go into the cytoplasm via something called the malate shuttle, but I'm not going to get into the details of that in this video. And then once it's in the cytoplasm, PEP carboxylase will then turn it into phosphoenolpyruvate. Phosphoenolpyruvate will then use the same enzymes that were used in glycolysis to get back to fructose 1,6 bisphosphate. We then need to use a different enzyme than glycolysis, and we use something called fructose 1,6 phosphatase. A phosphatase is an enzyme that removes a phosphate group, and that's exactly what this one does. It removes a phosphate from the first carbon of that fructose molecule. Notice that the phosphate is released as a free phosphate and does not create a molecule of ATP. Fructose 6-phosphate then turns back into glucose 6-phosphate using the same enzymes as in glycolysis. And finally, glucose 6-phosphate removes the carbon off of carbon 6 of glucose 6-phosphate to give us our final molecule of glucose. And in that process, another free phosphate is released. Earlier we said that PFK2 would have an important role in the fasting state, and gluconeogenesis is where it has that role. During gluconeogenesis, we are trying to create molecules of glucose. And those molecules of glucose are made in the liver but then sent to other cells, so it's crucial that the liver doesn't break down any of the molecules of glucose that it's making. The way that this gets achieved is that glucagon, which is released during the fasting state, leads to the phosphorylation of PFK2, and that turns off the kinase functioning of PFK2. And PFK2 is really unique because it actually has two functions, and one part of the enzyme can function as a phosphatase. And once it gets phosphorylated, the phosphatase turns on, and it removes the phosphate on the second carbon of fructose 2,6-bisphosphate, turning it back into fructose 6-phosphate. So glucagon is effectively lowering the amount of fructose 2,6-bisphosphate, which gets rid of the activating effect that molecule has on PFK1. So that, combined with the large amounts of ATP that's in the liver right now from fatty acid oxidation, we're going to be inhibiting PFK1, which is going to stop glycolysis and ensure that the liver doesn't use up the glucose that it's making. Remember what we said earlier about how most gluconeogenesis is not starting with a molecule of pyruvate, but rather some intermediate in glycolysis. And now we'll look at where do those intermediates come from. One of the main sources is glucogenic amino acids. A lot of these amino acids are going to come from the breakdown of protein in our muscles, but you can just think of it as from protein in general. They'll move through the bloodstream and then reach the liver, where deamination will occur. Deamination is the removal of an amine group from a molecule. And when we remove that amine group, we're transferring it to a molecule of alpha-ketoglutarate. So on the alpha-ketoglutarate, this carbonyl group is replaced with the amino group. And then the amino group on the glucogenic amino acid is replaced by a carbonyl group. And depending on the side chain of that amino acid, usually what it turns into is some Krebs cycle intermediate. That Krebs cycle intermediate will then go through the Krebs cycle and become a molecule of oxaloacetate, which, if you recall from earlier, is one of the molecules in gluconeogenesis that we can start with. After gaining the amino group, the alpha-ketoglutarate is now just glutamate. So this part here continues on as we use amino acids for gluconeogenesis. The glutamate will then transfer that amino group into the urea cycle, and that regenerates a molecule of alpha-ketoglutarate. That's really important to notice because a lot of people ask, why didn't we just use the molecule of alpha-ketoglutarate to make the oxaloacetate to then go into gluconeogenesis? The reason we don't do that is because we're not actually using up the alpha-ketoglutarate in this process because we regenerate it every time. So we keep using new glucogenic amino acids and using alpha-ketoglutarate as just a transporter of that amino group. And then the urea that's made will get excreted from the body in the urine. The second big source of glucogenic intermediates is glycerol. And we get glycerol mostly from the breakdown of triglycerides. Glycerol will make its way to the liver, where a multi-step process will turn it into DHAP, which is an intermediate in gluconeogenesis. Lactate, which is a product of anaerobic respiration, is our final glucogenic source. Through a reverse process in which we make lactate, NADH comes in, donates its electrons, and turns it right back into pyruvate. And that's also catalyzed by lactate dehydrogenase. Ketogenesis is the last part of the fasting stage that we'll go over. Ketogenesis is a way for the liver to create a molecule that can be used by other cells of the body as energy, and it helps to supplement the glucose that's being made by gluconeogenesis. That way the cells of the body that can't use fatty acids have two sources of energy, either the glucose from gluconeogenesis or ketone bodies from ketogenesis. What happens during ketogenesis, which takes place in the liver, is that the liver takes some of the acetyl-CoA that's being produced by fatty acid oxidation and turns it into ketone bodies, and here are two examples of ketone bodies. The ketone bodies then leave the liver and travel through the blood until they reach the target tissue. Once they reach that, they move into the cell, and then after they're inside the cell, they get turned back into acetyl-CoA. And then that acetyl-CoA can be fed into the Krebs cycle like normal. To recap all of this, I have the same screen that I had from the beginning of part 1, which is showing how we use fatty acids and glucose to create ATP. In the rest of the video, we talked about where these three molecules come from and what parts of the body are using them during the fed state, the first part of the fasting state, and the second part of the fasting state. During the first part of the fed state, every part of the body is using glucose that we got from our last meal. Once the sugar in our blood from that meal runs out, then glycogen breaks down into glucose, and the glycogen in the liver supplies glucose to the rest of the body, while the glycogen in the muscle is used by the muscles. So far in the fed state and in the fasting state, we don't use any ketones or fatty acids. After glycogen stores run out, adipose tissue starts breaking down triglycerides to supply fatty acids to many parts of the body. Some parts of the body can't use fatty acids, so the liver synthesizes glucose from gluconeogenesis and creates ketones from ketogenesis to supply those parts of the body with energy.